Hello friends, welcome to the third lecture, that is Centers of Diversity and Gene Banks. And one interesting thing I want to tell you today is today the result for ISP 2015 has been declared, and some of my friends have not been selected, and unfortunately I have also not made through this year, but I'm sure I will make through next year. So here I am with the lecture 3, Centers of Diversity and Gene Banks. Last lecture we had studied about domestication. It was very easy, not much to explain or to recap about. It's mostly theoretical. Now in this lecture again I'll try to make interesting by giving more and more examples. Let us first know what is genetic diversity. Diversity is mostly the variability, right? So it is the sum total of the genetic variation present in a population or a species. Not much to explain. Why is it needed? Why is genetic diversity needed? Because to develop better cultivars, to develop better varieties, we need a broad genetic base. We need to have a number of traits among which we can pick and choose. So we need to have a wide genetic base within which we can pick and choose, and of course wide adaptability. We have most of our wild relatives, they are very adaptable to a number of variety of environment conditions. So in order to develop improved cultivars, we need broad genetic base, that is greater genetic diversity. Now what can be the threat to this genetic diversity? It is something called as genetic erosion or extinction, and due to this we need to go for germ plug conservation. What is genetic erosion? See, in an already limited gene pool of an endangered species, a species which is already endangered, if it does not get a chance to breed or to multiply, to reproduce, its population will be further diminished. So this process is called as genetic erosion. A best example is China, where in 1949 nearly 10,000 wheat varieties were under cultivation compared to just 1,000 today. You can say less than 10% or nearly 10% of what was available in 1949. Some 50-60 years, 90% of the varieties have been eroded. Okay. Now this is something concept called as very important concept called as center of diversity and origin. What is center of diversity and origin, and are these two different or same? We'll study about it. Center of diversity is a geographical region with greatest genetic variability of a crop and its wild species. Not only a crop but also its wild species. Whereas center of origin is a geographical area from where they have originated. So in cases they may be coinciding, they may be same, but not always. And in both these areas, the plants have had opportunity to evolve resistance to various pathogens and other diseases via mutation. Sorry. Now, Vavilov, Russian scientist, postulated existence of 8 main centers and 3 subsidiary centers of diversity. And as per Vavilov, these centers of diversity were also their centers of origin. So, as per Vavilov, both of these coincided. And most of the centers that Vavilov had postulated, actually even today we can find a very high diversity in these areas. So what he noted, Vavilov, was that in most of these centers of origin, there were some common factors. One common factor was mountainous region in subtropical and tropical belts. Most of these areas occurred in the mountainous regions of tropics and subtropics. Another common factor was ancient agriculture and old civilization. with a very diverse ecotrophic topographic conditions, which is of course because in these areas when you find a great diversity of temperature, climatic factors, topography, it will give rise to more diversity of plants. These were the main centers: China, India, Central Asia, Asia Minor, that is Persia, Mediterranean, Abyssinia, that is Africa, Central America or Mexico, and South America. And then there were, as I told you, three subsidiary centers. One was Indomalia, second was Chile, that is again South American region, and third was Brazil and Paraguay. So these are the Wawillovian centers of genetic diversity, and these are some of the major crops. You can pause the video here. Basically, these are you can one thing that you'll find in common is with the last lecture is I had told most of these crops as the center of domestication against the places that are now showing as their centers of genetic diversity. So we can say that in the centers of genetic diversity also sometimes these crops were domesticated. But in case they were domesticated elsewhere, then the place where they were having most diversity was known as primary center, and the place where they were domesticated, that is other than their primary center, was known as secondary center. Okay, so these are again somewhat similar to the domestication chart that we had seen in the last lecture. Like soybean tea is common in China, India again rice, Central Asia and Asia you will find mostly wheat, rye, barley. Africa you'll find millets and coffee. Mexico you'll find maize, beans, gourd, squash. South America you'll find potato, tobacco, tomato. And Indo-Malaysia you'll of course find banana, coconut. Chile will be again potato. Brazil and Paraguay will be for pineapple, peanut and rubber tree. Now, as I told you in the last slide about primary, secondary and micro center. The classification in these three we can better understand by the way of comparison. Basically, primary center, if you just want to remember in short, is the center where there is maximum diversity of wild varieties, wild relatives. Okay, and secondary where there is maximum variety of domesticated species or domesticated variety. So, uh, and micro center is again a center within a center. Okay, so these are smaller areas with extreme or very high genetic diversity. Okay, these are the most important sites of evolution because the rate of evolution here is very fast. Okay, so all these are again very self-explanatory. Genetic diversity, of course, will be very high in primary. Secondary, it will be less than primary. Genes will be mostly dominant because I told you it was related with wild relatives, wild varieties. So mostly dominant. Crossing over will be less. Selection will be mostly natural selection because in wild varieties and in their natural habitat, mostly it will be natural selection. And because in secondary we are talking about domesticated varieties, so there will be both natural and artificial selection. Again, characters will be wild characters in primary and desirable characters in secondary. Again, I told you cultivation because these are wild varieties, so these are mostly in uncultivated areas. Secondary, these are domesticated varieties, so they are in cultivated areas. Simple. Now, this is one important term I want to tell you. It's just something to make it more interesting. Wawillovian mimicry or the weed mimicry. It's a form of mimicry where a weed comes to share one or more characteristics with a domesticated plant through generations of artificial selection. So basically what happens is in the process of domestication of a variety, we are weeding out the undesirable characteristics. But slowly and gradually, the weed also starts to develop those characters similar to the actual crop, main crop, for which the characters for which it is being weeded out. For example, we know we want bigger kernel size, grain size. We want varieties which should not shatter. We know we want varieties which should not be perennial, which should be annual. So slowly and gradually, the weed will also start developing these traits, favorable traits, so that along with the main crop plant, it will also get selected, and slowly and gradually, it will become another crop by itself, and it will come to be known as secondary crop. Okay. So basically, we have been doing the selection against the weed by killing a young weed or adult weed, separating its seeds. But to escape from being weeded out, weeds start developing similar characters to the crop plants. Like morphologically, their seeds become similar to the crop seeds, and thus they are unintentionally selected by the humans because they become indistinguishable from the main crop. So basically, they start mimicking the very character on the basis of which they could be weeded out. Best example in this term is herbicide resistance. In the recent times, we have been having glyphosate resistant crops, that is Roundup ready seeds, we are calling them, because we know it is a non-selective kind of a herbicide. So it is very cost efficient if we can develop seeds or crops which are resistant to this glyphosate Roundup. Then we'll not be requiring any other herbicide. But what has happened is because the weeds were being killed, they also started developing resistance to glyphosate, similar to the main crops, the genetically modified crops. So this is an example of Vavilovian mimicry. Now, I had told you, as they develop similar characters, they also end up being selected. So inadvertently, they are also being subjected to the pressures of selective breeding. So such Vavilovian mimics may eventually be domesticated, and then they are called as secondary crops. Best examples are rye, oats. Basically, these grew as weeds with wheat and barley, but came under selective pressures, and finally they developed as independent crops. Let's take the example of rye. Like wheat, it came to have a larger seeds and more rigid spindles to which the seeds are attached. But wheat is an annual plant, while wild rye is a perennial. But in some mutant varieties, we had the characteristics which were similar to the wheat plant. Like at the end of the growing season, wheat produces seed because it's an annual plant. Wild rye does not, so it is destroyed. But some mutants that set seeds are protected from destruction. Along with wheat, they'll be again resown. So slowly and gradually, it is being domesticated. Now, rye is a hardier plant than wheat. So in some of the areas, it is more suitable to grow rye. So having been selected through this process of wheat mimicry, it became a main crop in some of the areas where it was more favorable for cultivation of rye. Now, after studying Vavilovian mimicry, we'll study law of homologous series variation or law of parallel series of variation. Basically, a particular variation observed in a crop species is also expected to be available in its related species. For example, if we observe a dwarf variety in a particular species, it is also expected in other its related species. So this is known as law of parallel homologous series of variation. This principle was used by Vavilov as a clue for discovering similar characters in the related species. Now let us study about gene banks, gene sanctuaries. As we had told in the beginning of this lecture, that a threat to genetic diversity is genetic erosion. And to counter that threat, we have to go for germ plant conservation. Basically, on Broadly, the classification is in situ and ex situ. In situ is, of course, implying in the natural habitat. So, areas of great genetic diversity conserved in their natural habitat and protected from human interference. When we are protecting the germplasm in such condition, it is known as gene sanctuary or natural park or biosphere reserve. They are basically established in microcenters where there is high rate of genetic diversity. First one in India is in Garo Hills in Assam, and it is for in situ conservation of citrus. Basically, it protects the loss of genetic diversity which can happen by human intervention, and it also allows natural selection and evolution to operate. There's two main drawbacks: entire variability of species can't be conserved, and secondly, its maintenance and establishment is a very difficult task, and it is a very expensive process. Now coming to HC2, germ plasm banks, gene banks, or we will see the three types also. It is basically a place where germ plasm can be conserved in a living state, whether it be in form of seeds, pollen, in vitro culture, or in form of field gene banks, in form of like living plant sample growing in the field. So mainly three types on the basis of, we can say, seed banks, field gene banks, and meristem gene banks. Let us have a look one by one. Basically, seeds are easy to store because they occupy lesser space, and also they can be dried. Some of the seeds can be dried and stored at a very low temperature without losing their viability. Such seeds are known as orthodox. Others which lose their viability are known as recalcitrant. So such way recalcitrant species cannot be stored in seed banks and require in situ conservation. Now there are three types of seed collection. One is base collection, another is active collection, and third is working collection. Basically, it is on the basis of how frequently or how actively these collections are being used in breeding. Active, as you can say, more, it is used more than the base collection, and working collection it is used most frequently out of the three in breeding programs by the breeders. So again, based on these three types of seed collection, there are three types of conservation: long term, medium term, and short term. So for base varieties, base seed collection, the method is long term, and how much long? It is for 50 years at minus 18 to minus 20 degrees C. For active, again it is medium term, 10 to 15 years at 0 degrees C. And working, again it is, you can say, the temperature is going higher and the period is decreasing. So short term, and that is 3 to 5 years and at 5 to 10 degrees C. Of course, we can apply the logic that because working collection we are using most frequently, so we don't need a very low temperature because we are going to be using it very frequently. So now what are the merits and demerits? First of all, again, easy storage. Second, easy handling. Third and fourth, it is conservation from pest and disease free environment, and cheap, and it's also possible with the help of seed banks to store entire viability at one single place because seeds don't take much space. Demerits are, we cannot have recalcitrant varieties in seed banks, and also power failure may lead to loss of entire viability of the seeds because we are storing at a very low temperature. For that we need power. And third is required periodic evaluation of the seed viability because you have to keep on checking whether or not they are active or the seed is viable or not. So after some time, multiplication is inevitable to get the fresh seeds for storage. Okay. Now the second category we had seen as field gene banks or plant gene banks. Here it is, germplasm is maintained in living in form of living plants, in form of permanent living collection, in form of growing plants. Basically, it is, again we had seen, it is for the plants which have recalcitrant seeds or which do not produce seeds at all. So basically, it is used to maintain a working collection of plants. Sorry, it's not plants, it is plants for experimental purpose. So it is, we can imagine very well what could be the examples: coconut, rubber, mango, yam, cocoa. These are the plants for which we are using field gene banks. Later on we'll see the examples where these are located. Main advantage of field gene banks is it can be directly utilized in the breeding program because we are having live collection of plants. Also, it is giving opportunity for continuous evaluation for economic characters because we are having live plants. And but the main disadvantage is it is costly and it does not cover the entire variability because it is, again, it takes up space and all the cost. So also, the germplasm, if we are going in the open in the natural condition, it will be exposed to pests and diseases and other natural calamities. In the case of seed banks, it was protected from all these. These are some of the areas. Internationally, we have field gene banks for oil palm in Malaysia, for coconut in Indonesia, and India we have again for coconut in Andaman Nicobar. Philippines we have for banana. These are some of the field gene banks in India. Wheat we have in Karnal, that is Directorate of Wheat Research in Karnal. Rice, Cuttack, CRRI. Potato in Shimla, CPRI. Cotton, CICR in Nagpur. Pulses in Kanpur. Oilseeds in Hyderabad. Again, these are all you have to mug up. Sugarcane is in Coimbatore. IGFRI, most many times it is asked in the objective question. CTCRI, that is Center for Tuber Crops Research Institute, and IIHR Hisarghatta, that is specifically in Bangalore for horticulture crops. Grapes in RCG, that is in Pune. Third and last type of gene bank is meristem gene bank. Again, we had seen one seed, second was field gene bank, another third is meristem. So seeds which are orthodox can be stored through gene bank. Recalcitrant and other which do not produce seed at all can be stored through the field gene bank. But such asexually propagated species, such varieties cannot, such species can be stored through meristem gene bank. Like that is in tissue culture. We know what meristem is. Mostly for horticulture species it is used, and again it can be used in two ways. First, we have to we can either store under slow growth conditions, or second, we can store for long term under cryopreservation, that is at extremely low temperature, minus 196 degrees C at in liquid temperature, and virtually all the biological processes stop at this temperature. So this was meristem gene bank. Now coming to pollen and DNA, it is very rare, and why it is there? It is a very elaborate process and also very expensive. So that is all. We have finished the third lecture. We have seen about the centers of diversity, centers of origin, Pavlovian centers of diversity, Pavlovian mimicry, homologous series of variation, okay, and gene banks and gene sanctuaries. So that is all. Thank you.